Hello everyone and welcome to the NanoReach podcast, a science outreach initiative of the Nanomedicine Research Group here at ICT Mumbai. I'm your host Anushka Chaudhary, a science communicator with the NRG, and joining me today is Dr. Clive Roper, currently a director at Roper Toxicology Consulting, wherein he helps companies strategize their scientific needs to move towards non-animal technologies in toxicology testing. He is also a vice president at the European Society of Toxicology in-vitro and a member of the board of directors at the North American 3Rs Collaborative. So it's a pleasure to have you here on our podcast.
Thank you very much.
How are you doing today?
Very good.
Yeah. So is this your first time in India?
No, I've been to Goa, but maybe 23 years ago. So So this is really my proper time in, first time, certainly in a long time, and it's wonderful.
How are you finding Mumbai?
It's fast, it's fun, the smells, the sights, the food, the tea.
The food.
The food. It's wonderful. It's so different from anything that we ever experience in Europe. The people are so friendly. It's I feel I'm almost treated like a king here. There's people are wonderful, and the food, the drink, the cups of tea. It's wonderful to be here, and I'm thoroughly enjoying the conference as well.
That's really good to hear. As you're the director at Roper Toxicology, so what is the primary research focus of this company?
So my business has been going for nearly 3 years, and I'm a consultant. Everything that I do is in vitro toxicology. So I work with on-a-chip companies, helping them to meet potential customers. I also look at their test methods, see if there's any ways that I can help them improve reproducibility. I work with multinational corporations who are looking to find new ways of testing. So if you may be aware that in the past 30 years, there's been no change in the failure rate of going from preclinical to clinical.
Yes.
It's still about 90% failure rate. So we do more and more animal tests, and we don't improve that. So there's a real interest in moving to human in vitro tests. So I help them to find the different tests that might be available, or I might help them to find companies that perform similar tests, and then we can innovate to generate the right type of tests that they're interested in. I work with biotechs, and then I spend a lot of time supporting new research, new scientists with our through European Society of Toxicology in vitro. And then my bigger picture observations is supporting the UK NC3Rs, which I'm also a director of, and the 3Rs Collaborative in the US, which I've been with since it started 6 or 7 years ago. So
it's really interesting. So what got you into in vitro toxicology and the overall area of moving to non-animal methods of testing?
Well, we have to go back to 1990.
Sure,
we've got to go back a long time. So I was quite, I would say I had quite extreme views about the use of animals, and I thought, well, can I go there and be a demonstrator? Shall I demonstrate about the use of animals?
Be an activist.
Shall I be an activist? And I thought, doesn't seem to make much sense, because that's being really aggressive. If you have a group who says no and another group who says yes, well, there's no way you can have dialogue.
Yeah. You're not actually moving forward.
No one is benefiting. No one. So the activist doesn't understand why the animal is being used, and the animal users don't understand why they would want to communicate with people. So I decided that I would do something about it and start to work in the area of replacement of animals. So I started a PhD funded by Unilever Research at Newcastle University in England, And the whole purpose of that actually wasn't animal replacement. It was to generate a test that we could create that would be useful for looking at absorption of chemicals through the skin. And our secondary and then if that's successful, then that can be used in when they put the ban in place, in the cosmetic ban, then we would have a test that we could use. Now, the consequence of that has been over the past 25 years, we've increasingly replaced the use of those tests in chemical testing. That's now replaced in ECHA.
Yeah.
We've replaced it in cosmetics. And in pharma, we've got the FDA IVRT, IVPT. And then the most recent one was that we ended at with the EPA, ended using that 3 or 4 years ago, and I was involved in that as well. So although it's taken maybe 25 years, we've effectively stopped the use of the in vivo skin absorption test and replaced it with the in vivo test. 25 years might sound a long time, but we're learning our way, and now we're finding new things. So I started doing respiratory tox about 2012, and now we got our first success with a full replacement with the US EPA of replacing a 90-day rodent repeat dose assay. But our objective wasn't to replace that. It was to see if we could actually get enough information from human in vitro, and we did. We got the waiver for the 90-day, because that was that wasn't our objective. It was to get the data that allows us to protect humans. If you've got the data, you don't need the animal test. And now we've written that up as an OECD case study. Number 367, do have a look at it. We're really proud of it.
That's quite an achievement.
And so we've gone from say 2012 we started that program. I think it was about 22 that we did publish the data, publish the case study, and it was about 2018, 2019 that we did got the risk assessment done. So we're increasingly getting better at it, and the time is now getting shorter and as we're getting better at it. And also there's a huge interest from regulatory agencies, all the stakeholders, companies, that they want to be able to get better data to protect humans and to protect animals. So I think that the choice that I made in 1990 to be generating tests rather than being an activist, I'm pleased.
It's a really good choice that you made, because it led to a lot of great initiatives in this field.
And I know a lot of activists who are now learning about what we're doing and supporting us. And now there's a lot more dialogue going on between the different groups, and I think that that's really positive. I'm working with groups who I would find quite difficult in the past, and I think they're absolutely wonderful. They're helping us to take away the politics as we look at the science. So even that has changed, and I think that the more we have dialogue, the more we can progress to get better tests, and we'll get better drugs on the market as a consequence.
Yes. Because overall the timeline reduces when you move on to in vitro tests.
Yeah. And I always focus on, when we talk about in vitro, I always say, well, in vitro human.
Right.
And there's a lot of really good in vitro animal models, and they're very useful for validation, because if we do an in vivo test, it's in an animal, we do an in vitro human test, well, that's got 2 steps of change.
Yes.
And traditionally we've gone and said, oh, the results aren't the same, but of course we've got a species difference and in vitro, in vivo. So if we can now add in human in vitro, animal in vitro, animal in vivo, we can compare those, and that's what we did with the US EPA to end their use of the in vivo test for skin. What they saw was that the in vitro rat was more conservative than the in vivo rat in general, that meant that we would say that the human in vitro would be more conservative than the human in vivo, so we're being protected. So you only need to really perform the in vitro human for submissions to the EPA. So that's been a really exciting process. We're now, when we're validating, if we've got a human and if we've got an animal in vitro test and an animal in vivo test, then that's great because they should be similar. If they are,
then
and we have something different human in vivo, if our human in vitro is different, well then we know that's a species difference, and so we can have reliability, and so this confirms. I mean, it's an expensive way to do it, and we don't always need to do that, but there are a lot of cases. And there's also a really useful thing about having an animal in vitro, because there's still a lot of, we can't replace all animals yet, but we can actually get information to make decisions to give animal welfare. So if we can use an animal in vitro model, and we've got nice ones for the lung, that allows us to replace, to make some decisions. If we see lots of toxicity, we'll choose not to run that test. So it's an indirect replacement, and it allows us to give better animal welfare. So there's a lot of good reasons to run these complex sets of models, and I think we're in a really exciting time where we've got lots of great science, and goodness, I've seen some incredible science while I've been here in India for the past only a week, and what I've met with the academics here at the ICT, the students, the PhD students are doing things which are just unbelievable. And I've seen some great labs which I would be surprised are in India, but they've got wonderful quality. I mean, I'm really impressed with what I've seen here in India, and I think the future's very bright for Indian science.
We've made quite a lot of progress over the years in terms of funding our research.
Yes.
And motivating students to actually pursue research.
I don't think they need much. The guys you've got here don't need much motivation. They're hugely enthusiastic about what they do, and they've been telling me about what their projects, and have been asking me for advice, and we've really gone into some quite detail about some incredible projects. So I've heard a really nice project in the ICT on age-related macular degeneration.
Yes.
It's a fantastic model there, which is not far away from being commercially available, I think. I mean, it's really exciting. Running some new skin models, some more complex in vitro skin models. Yeah, and the students have been so fascinating about what they're doing. And I think that they're the quality of the science, the quality of the product that they're doing is incredible. So, and this is just one institute. So
speaking of motivation for research and passion for research, what made you take up research in academia or science as a career?
I can actually remember back to being about 8 years old, going into the cupboards and mixing chemicals together. And I decided I was going to be a professor when I grew up. Well, that didn't happen. But I've gone into that area of science and gone into the industrial side of science. But maybe one day the hope will become perhaps still to become a professor. But you know, you follow a path, and I think that fire was always with me to be a scientist. And the route was in industrial science, but I've thoroughly enjoyed every minute of my career. So it's been a wonderful career, and it's still a few years to go.
A long way to go. Still a long way to go.
Thank you.
Moving on to not being dependent on animal models so much. So in the next 10 years or so, where do you see this industry going? What is the scope? Will we actually completely bypass animal testing? I don't think we're going to fully remove animal testing. I think there's still a lot of things that we can't do. But I think there is going to be an acceleration. I mean, if we look in drug development, if we look in the drug space, traditionally we used small molecules. And, you know, when you move to large molecules or new modalities, we're dealing with bypass animal testing?
I don't think we're going to fully remove animal testing. I think there's still a lot of things that we can't do, but I think there is going to be an acceleration. I mean, if we look in drug development, if we look in the drug space, traditionally we used small molecules, and you know, when you move to large molecules or new modalities, we're dealing with the biology is very human specific. So if you run, if you put so a lot of these are proteins, you put them into an animal, the animal will see that as a foreign protein, chop it up. So you see there's no efficacy, there's no toxicity. And of course we saw that with the Northwich Park incident maybe 10 or 12 years ago, where there was a large molecule tested normally, and when they brought it into humans there was an adverse event, and it was probably about 50 to 100-fold too high concentration. I mean, now we would pick that up, we've got other ways of doing it, but the more we're seeing a lot of these problems, we're putting them into these molecules, we're putting into animals, nothing happens. We're not seeing toxicity. There might be an NHP version and a human version and a mouse version. So those versions are maybe too specific for the human. So I think that we have to show safety and efficacy, and we really got to start moving those cases to human models. Some of them might be very simple. They might be 2D models. They might be tissue models, and of course they might be very complex organ chip models. And as we've seen in the past few days of this workshop, we don't have to have a super complicated, expensive organ chip model. There can be quite simple models. So I think that we're increasingly going to find the benefits. We're going to get better molecules into the safety program, because we're going to be using more human in vitro testing in the screening phase. That means we're going to get better molecules going into our clinical trials, and that means we get better molecules or better, even the new modalities, better treatments to the patients who are desperate to deal with these diseases. So as I say, very exciting to see what you're doing here with the AMD, because it's an unmet need. The animals don't work. The eye is too different in mice, for example. They don't have cones. They've got a different structure. So actually, if we can generate a human in vitro model, and it looks like you're doing it, that we can use as a drug discovery efficacy evaluation, we can get better molecules to an aging population. So AMD is going to increase. We're going to get more and more as we get an aging population. So if we can stop that, I mean, so we might have an aging population, but we're also we need them to keep a good quality of life. And if they're losing sight, their quality of life is reducing. So what you're doing here is fundamentally important in getting drugs to this increasingly problematic disease that we have with our aging population. So fascinating what you guys do. It's been an education. I came here I certainly came here to share my knowledge, but also I came here to learn, and I would say I might well be the benefactor. Well, I mean, it's just been non-stop people telling me about what they're doing, asking me for advice, telling me about what they're working on, and and it shouldn't be a one-way traffic. I mean, I'm here to learn as well. So, but I've learned a huge amount. So, thank you very much.
Thank you so much for being on the podcast. It was great to hear your experience. And in the end, would you have any message to young researchers out there?
It's getting bigger, it's exciting, the opportunities for breakthroughs, some even small incremental changes will go from being a model that looks interesting to a commercial model that's going to help save lives and improve the quality of life of people. So come here and do research. Come here and bring new models to market. Come here and find out the wonderful science here in India, and And I've worked in US and UK. I've worked primarily in UK, but I've been to many places in Europe and in US. Fabulous here. I mean, it's India has got great quality, and I think in Europe and US we need to continue to partner with our Indian friends.
That's really great to hear. Thank you so much for being on the podcast.
Thank you very much for inviting me.
Thank you for tuning into this episode of the NanoReach podcast. If you like this episode, please share it with your peers and subscribe to the Nanomedicine Research Group on YouTube and Spotify for more such episodes. Follow us on Instagram at nrg_ICT Mumbai for regular updates. Thank you.